Good morning. I want to make sure that everybody who's sitting down has taken a peanut. There are bags at the entrances to the room, and also there are bags in the middle part of the room and in the side rooms. For those of you who are not here for the peanut demonstration, you're going to miss an important lesson which will likely be reappearing perhaps on the midterm. So it's too bad you can't go with us with this demo. Today, what I want to do is to, first of all, recap for you some of the important points of the last lecture on angiosperms, and then secondly, to proceed to discuss the angiosperms, which, as I said before, are going to be the main topic or the main plants that we're going to consider for the remaining portion of the plants section of the course. Now, last time we said that the angiosperms came to be a dominant part of our flora. They are the dominant species in our flora, except at the very high altitudes, because of the development of a number of unique structures. And the first was the fruit, which we said in many cases, in all cases, consists at least in part of the ovary, or really the ovary wall. And we also said that the angiosperms were successful because they developed a flower, or flowers, which were the means of attracting a pollinator. That is, some organism other than the flower or the wind to move the pollen from one plant to another and thereby affect fertilization, to have the sexual life cycle. So these are two extremely important characteristics that the angiosperms develop, and they are major ones that account for the dominance of our flora by angiosperms. Today we'll talk about some additional characteristics, although they may not be quite as important or significant as these two. So with today's lecture, what I want to do is to begin to talk to you about development of the angiosperm plant body. And in particular, we're going to begin today talking about the physiological and biochemical controls of development in plants. And this topic, or this theme, is going to occupy most of the lectures as we go through for the rest of the portion on plants. Now, for a discussion on the controls of development in a plant, we have to know something about plant structure and also something about the internal part of the plant, the anatomy. Plant anatomy, unlike animal anatomy, is simpler, but yet there are some specialized cell types that I want to introduce you to. Most of you have familiarity with these cell types, although may not realize where they were or what they were in the plants. Now, when we start to talk about plant development and the controls of plant development, a theme or a point which should be in the back of your mind is that once the plant germinates and sends down its roots in a particular portion of the soil or maybe growing on a rock, that plant is essentially committed to spending its entire life in that spot. If the weather turns bad or water or nutrients are low in the soil, that plant cannot get up and leave. It can't move like an animal would to a new area where the environment was more conducive to the growth of the animal. So plants, because they are essentially rooted for their entire lifetime or fixed to their environment, and these are the higher plants like angiosperms and gymnosperms, have developed a number of mechanisms for allowing them to continue to prosper and live because they are fixed to their environment. And the first mechanism which they've developed, and we'll spend some time talking about it, is they have developed for mechanisms for sensing, or you could say measuring the environment. And although we don't have time to talk about it in this course, these mechanisms are exceedingly sensitive, much more sensitive than mechanisms that we have. The second mechanism that they have, which allows them to prosper, although they are fixed to their environment, is that they have a wide array of developmental mechanisms which allow adaptation, which allow them to adapt. So these are sort of themes that are going to be underlying a lot of the discussion which we have. How does a plant sense the changes in its environment, and then how does it respond in a physiological or biochemical way? Last time when we ended the lecture, we ended by talking about the angiosperm seed. And we said that as a result of double fertilization, within the seed, two types of general tissues formed. And those tissues were the embryo, which is diploid, and a second type of tissue, which we said we call the endosperm, which is 3N, or triploid. And today, what I want to do is to essentially continue talking, and then we would have the seed coat on the outside. Today, what I want to do is to follow now the development of the embryo as it gets to a state of maturity, and then to talk about the germination of the seed and the establishment of the new plant, the new generation. Now, in angiosperms, if we were to dissect seeds and open them up when the embryo was mature, we would find that the embryos take one of two forms. And these two forms allow us to divide the angiosperms into two generalized groups. So let's take a look at what the two embryo types look like. In one embryo type, if you were to dissect the embryo out, you'd find something like this. And this is a kind of a cartoon. There are better pictures in your book. This would be the axis. This would be the embryo axis. And on one end of the axis, you'd find on the outside two bumps. Call them bumps. So one category or one division of the embryos would have embryos that have two bumps on either side of the embryo axis. These two bumps are known as seed leaves. And they're called seed leaves because they're in the seed. And the technical name for these are cotyledons. Cotyledons. And because this particular type of embryo has two bumps, we call it di for two, and we shorten the word cotyledon to COTS, C-O-T-S. So the old name for plants which showed, or the grouping for plants which showed these kind of bumps on the embryo axis, were called dicots. And now that name has been modified slightly, as you will see from your textbook. We add the word U to the front of it. So plants with two bumps in their embryo are called eudicots. And this is contrasted now with an embryo which doesn't have two bumps. So let's draw an embryo axis where we have essentially only one bump on the axis. And let's just do it like so. In this particular cartoon or picture, the bump is over here. The bump is over here. It's kind of placed to the side of the axis. And there's only one. And plants which fall into this division of the angiosperms that have only one bump on their axis, and this would again be the axis here, we call these plants monocots or monocotyledonous plants. But monocots is the short term for them. Now, what I'd like to do is to talk to you about the seeds which have these two types of development, and specifically to focus on what happens to the embryo and what happens to the endosperm, this sort of strange triploid tissue that we've introduced you to. In the beginning, if we look, and let's say we're going to look now at a eudicot seed, and let's say it is mature. So we're going to take that seed that I drew now, make the seed coat on the outside. If we were to look at this seed, you recall the seed originally had inside of it an embryo and a mass of endosperm. In the typical eudicot seed, the embryo fills much of the seed. So this would be the embryo. And the endosperm is much reduced or absent. I shouldn't say absent, or you can't see it. Much reduced or not detected. So the typical situation in most eudicots, not all, but most eudicots, is that the embryo grows to fill the entire space inside the seed coat, and the endosperm, if it's present, is very, very much reduced. In the monocots, the typical, and this is a generalization, the typical situation is quite the reverse. So let's make a monocot seed. Monocot seed. Here we would have the embryo over to the side of the seed, embryo over to the side, and then we might have the single cotyledon. So most of this seed would be filled with endosperm. And the embryo occupies a very minor portion of the total volume of the seed. Now, what do the endosperm and the cotyledons do? We're making a big point of their importance for the seed. What we can say is that endosperm and large cotyledons function in providing nutrients to allow the seedling to become established. All of you are very familiar with these materials, and we're going to talk about them more. The cotyledons are foods that many of us eat. We're going to talk about them, and we find them nutritious for the same reason that the developing seedling finds them nutritious. There's a large amount of nutrients in them. We're also going to talk about the endosperm, which provides nutrients, and we as humans in many forms use the nutrients in the endosperm, and I'll talk to you about some examples in a moment. Now let's look at the embryo axis shortly before germination. So we're going to look at the mature embryo axis, maybe just before germination. So here is the axis of the embryo. This would be the axis. The axis of the embryo. And we'll put the cotyledons on. Let's say this is a eudicot, so these would be the cotyledons. Now, of all the points that I'm going to make in today's lecture, probably the most important, and the one which is also the hardest for you to kind of grasp, is the following. And I'll write this down for you. At either end of the axis, are zones of high cell division activity. Plant biologists call zones of high cell division activity, we call these meristems. Zones of high cell division activity. So at the top or the shoot end, we have a meristem, and that meristem is the shoot, and because it's at the tip, apical meristem. So we say we have a zone of high cell division activity at the shoot, tip of the axis. Similarly, at the bottom of the axis, where the root would form, we have a meristem. And because it is at the tip where the root will form, we call it the root apical meristem. Now, this is the conceptual part, which is probably the most difficult. And I want to write it down and say it slowly because it's the most important point probably for today's lecture. If a new cell or tissue can be traced in its origin, or can trace its origin directly to the cell division activity of the meristem, we call these new cells or tissues, or we characterize these new cells or tissues as primary. So they are primary cells, primary tissues. That is, through the cell division activity at either the shoot apical meristem, through the mitosis there, or at the root apical meristem, we have produced new cells or new tissues. And if those new cells or new tissues can directly be traced back in their formation to the mitotic activity at these two meristems, we call those new cells or tissues primary. And it's important that you understand what primary means, because as you might guess, there's going to be a secondary, and that has to be something that you can distinguish easily in your mind. Now what I'd like to do is to talk a little bit about the other tissue, other tissues which make up the embryo axis. We call this tissue, which is directly above the cotyledons, and the reason I'm giving you these two terms is because we're going to refer back to them. We call this tissue above the epicotyl, the tissue that is above the cotyledons, and the tissue which is below, going down to the root, we call it the hypocotyl. So this is just to define two different regions of the embryo axis. In monocots, and I have a good example of a monocot here, which is corn, that most of you are familiar with, we said that the embryo occupies a very small or narrow portion of the seed of the corn. That most of the corn seed, and these, as you will find out when we have labs, are really, each seed that you're calling a seed is really a fruit, because it's an ovary we're looking at. But let's say for the purposes we'll call it a seed. Each of these seeds has inside of them a small embryo. That's the thing that gets stuck in your teeth when you eat corn. And most of the inside of the seed is filled with endosperm. Now, those of you who are who grow corn yourself or who get it fresh from a market, say like Monterey Market or Berkeley Bowl, know that if you bite into an ear of fresh corn, you inevitably get squirted with liquid. That liquid is endosperm. That liquid contains millions of triploid nuclei that are floating around in this liquid that provides nutrients for the seed once it germinates. Now, let's talk about what happens to an ear of corn if a human doesn't interfere with it. Here the corn forms on the plant, and it makes liquid endosperm and an embryo. That liquid endosperm, as most of you know if you've had fresh corn, is sweet, high in sugars. Let's say this ear now falls to the ground and remains on the ground for a long period of time, and winter comes along. When the ear falls on the ground, the liquid endosperm, which contains large amounts of sugars in it, begins to be converted to starch. And this is what happens if you buy corn which has been sitting around for a long time. The liquid endosperm now has been converted from a sweet material to a starchy material. So this ear falls on the ground, starch is now being formed, and it remains on the ground all winter. And then springtime comes along, and in order for the embryo now to use the nutrients that are stored in the endosperm, that endosperm has to be reconverted back to sugar again. So when corn seeds germinate, if you pick them out and you taste them again, if you're willing to do that, they'll be sweet again. But in between, they're starchy. So a question I want to ask you is, why would this plant, in its natural system, natural environment, go through the energy-demanding process of first making sugar, which is, of course, a product of photosynthesis, then converting it to starch for a period of time, then converting it back to sugar again when germination occurs? What possible advantage in the life cycle of this plant could it be to do this conversion process, which is energetically quite demanding? Another example of endosperm that most of you are familiar with, at least familiar with as far as a phenomenon, but maybe not realize it, is coconut milk. Coconut milk, coconut is essentially, this is a monocot seed, and part of what you see on the outside is the fruit wall. For those of you who open a coconut up, and I'll show you a coconut in a moment, when you open it up, the white material represents endosperm that has now begun to cellularize. So in each of the cells, if you could look at them in the white material of a coconut, each cell making up that white material would have a triploid nucleus in it, and it would begin to solidify. But when you buy fresh coconuts, there's milk, as we call it. This milk is liquid endosperm, millions of triploid nuclei floating around. And for those of you who are interested in sort of particular organic lifestyle or very healthy foods, the liquid endosperm contains, so far unanalyzed, or not completely analyzed, huge composition of nutrients. It contains a vast array of amino acids and proteins, things that we have not begun to analyze. And you can buy coconut milk in the store. Coconut milk, as I said, represents a rich supply of so far uncharacterized or incompletely characterized nutrients. Now, if you open the coconut up, and this is a coconut fruit, and we're going to talk about this a little bit more. This one is a hinged coconut, which we've cut open. You actually can see the endosperm, the white material that you eat, beginning to solidify in the coconut. Around the outside, we have the fruit wall, and this would represent, this hard stuff on the outside, the fibrous stuff, would represent the inner part of the fruit wall. And the seed coat would be the brown material that you frequently see when you cut out the coconut meat. Now, this endosperm is so rich so that when the coconut is ready to germinate, it is carrying with it a large amount of material. And what I want to do now is to open up one of the coconuts and to show you where the embryo is in relation to the endosperm, which is around the outside of the coconut. So typically, if we take a coconut, and let's pretend like we're going to take this coconut here and cut it in half so that you're looking at it in half, what you would find is here is a section of a coconut. What you would find at one end is the embryo. We'll talk about that end in a moment. Then you'd find the cotyledon. Then you'd find liquid endosperm. And around the sides, as we mentioned before, you would see the white so-called meat of the coconut, which is solidified endosperm. Now, when you go to the supermarket, next time you're there, or if you want to come up after the lecture, we'll have these in the class, look at the end of a coconut. A coconut has, as many of you know colloquially, these things that are called eyes. It has 3 eyes on the bottom. As you will find out when we talk more about the monocots have their parts in threes. There are parts in threes. Each coconut represents 3 carpels that have individually fused together. Remember, a carpel contains inside of the ovule, which ultimately will contain the seed. Each coconut represents 3 carpels, and you can actually see the lines where the carpels have fused together. And normally what happens is that the single ovule in each coconut is fertilized. So the plant now has 3 developing embryos inside of it. And at some point, the plant says, I can only support one of these embryos. I want to put all my nutrients to that one embryo. So it aborts the other 2. The one that it keeps going becomes the larger area, or the larger quadrant of the coconut. It is the biggest eye. So if you open a coconut up and you look underneath the largest eye, you'll be able to find the embryo. So for those of you who I told to bring umbrellas if you sit in the front row, this might be a good time to open the umbrellas. For those of you who don't have umbrellas, I have a surprise for you. I already have a coconut that I've opened, so let me bring it up here. But if I didn't, what you would want to do is to hit the coconut on the end where the 3 sutures come together, and be particularly careful of where you don't have. I'm not going to do it, I promise you. Although it would sound good. Where the 3 sutures come together. And now we're going to look under the eye, which was the largest eye of this coconut that I have here. And the embryo is here. And I'll leave it out for you to see. I'm going to show you a picture of it in a couple of seconds. So here we have a coconut where we have opened it up. If you're ever at a boring party sometime and they have coconuts and you want to spice up the party, dissect out the embryo and people will never think of you the same way. You'll always be invited back. All right, so let me show you one of these coconuts now. I don't have to have this on. Here is a coconut that I've opened up, and I managed to make sure that the big eye was not injured or destroyed. So I'm going to open this coconut up, and in this particular coconut, you can see the white meat, which represents the solidified liquid endosperm. I've already drained out the liquid that was still present inside of the coconut. And if you look carefully, and I'll show you a picture of this in a moment, and I'll leave it up for the lecture for you to see, you can see the small embryo, which is about 3/4 of a centimeter in length. It will eventually, it's cotyledon, just like we've drawn over here. This cotyledon will eventually enlarge and grow into the endosperm and absorb the nutrients and use the nutrients to establish the new seedling. And we'll talk about this a little bit more. Now, dicots, or eudicots, also have endosperm. And we said that endosperm and embryos form simultaneously in all of the angiosperms as a result of double fertilization. But in general, in angiosperms, which are eudicots, the endosperm inside the seed is reduced or lost. And that endosperm doesn't just go away or disappear. Its nutrients are absorbed into the cotyledons. So a typical eudicot seed, such as this walnut seed, if we open it up and look inside of it, the so-called meat that you consume of this seed, this meat that you consume, these are the cotyledons of the walnut. And the nutrients which were present at the time the embryo was forming and enlarging were in the endosperm, and they are gradually absorbed into the developing embryo. Now, I've given all of you a peanut. I'd like you to take the peanut out for a moment and have a look at it. We're going to crack it, and these aren't roasted, so they probably won't be very good tasting, and probably good if you could not throw the shells on the floor. If you open the peanut up, before you open it up, on the outside, what do you think that structure is on the outside? Most of you think it's the seed coat, but in fact, it's the ovary wall. So in this particular case, the ovary has become hard and dry. And now if you go inside of it, you will be able to see sort of brown papery covering for each of the 1 or 2 or 3 seeds that you have inside. That brown papery covering is the seed coat. And notice in this case the seed coat is very fragile. Now if you take an individual seed and you split it in half, you're dividing that seed into the two cotyledons. So when you eat peanuts, you are eating the two cotyledons of the peanut. And if you take that seed now and you look at one end of the seed, what you will find is that there's a tiny outgrowth. That tiny outgrowth represents the embryo axis of the seed, and when the peanut seed germinates, the nutrients that are stored in the cotyledons are channeled or moved into the developing embryo axis. Let me show you now a few pictures of the coconut that we talked about. Here is the coconut, a typical coconut that we have up here in front. And what I want to point out to you is here is the large eye. It occupies the largest quadrant of the coconut. And here are two smaller eyes. They represent probably coconut embryos that have aborted. And now if we take a typical coconut and we hit it right on this ridge here, often the crack will go right through the middle of that large eye, which I have up here in front for you to see. And to show you what the embryo looks like, here is the embryo. This would be the cotyledon end of the embryo, and this would be the shoot end of the embryo. So the embryo would be facing into the coconut in this direction. And finally, if you allow the coconut to begin to germinate, all of the liquid endosperm would eventually solidify. And the cotyledon would grow into that coconut. This is obviously a coconut which has been cut in half. And it would then be used for nutrients for the developing seedling. So this is the coconut. Now I'm going to leave the screen down, Dave, but could we just have the lights for a second? That would be good. Let me go on to the next one to get this out of the way. Oops. All right. Let's see that here. We'll just close that. Okay, perfect. Now, the other factors that I want to talk to you about, which involve seeds that have made the angiosperm such a good and successful group, we mentioned the fruit, we mentioned the flower. The next factor that I want to put in, which really helped the angiosperms develop into such great organisms, is the fact that they develop mechanisms for dispersing their fruits and their seeds. And what I want to do now is briefly talk about mechanisms for dispersing fruits and seeds. Some of these are very simple and straightforward mechanisms. For example, the wind is often used to disperse seeds. And wind tends to be used in seeds that are small, such as the seeds of an orchid. They can be blown great distances. Many of you have seen dandelions. Many plants develop sort of devices which are like parachutes, and I'll show you a picture of them, to help them increase the surface area to be pulled away by the wind. For those of you who are from the Southwest, you have a really good example of dispersal by the wind, but it isn't dispersal of the individual seed. It is dispersal of the whole plant. So for those of you who have seen tumbleweeds, here is a plant which essentially is cut off where its shoot joins the ground or joins the root, and as the wind pushes it along, as it tumbles the weed along, it disperses its seed. So this is a very good example. There are some plants which actually shoot their seeds off, and we're going to show you some examples of that in a moment, where they have a real mechanism. If you touch the plant, or if an animal walks by and touches it, the seed is rapidly shot off, often into the fur of the animal, and then it's carried someplace else and dispersed. There are also plants, and this is probably one which is familiar to you, which are dispersed by water. This is unfortunately a dead coconut plant, but nevertheless, not only is it dead, it doesn't go to the pot. Here's a plant in which the coconut fruit is modified for floating. So this plant is dispersed by water, and it carries within it, as you know, the liquid endosperm, which is a package of nutrients. So water is also used. If a seed is at the top of a hill or near a creek or a river, you can imagine how that water would disperse the seed. Many seeds and fruits are eaten by birds, and this is the reason why seeds and fruits often are packaged in very sweet aromatic, colorful sort of packages, which are the fruits. They are meant to be eaten by animals. The animals eat the fruit and then fly away and defecate the seed in some other area. Often, it is necessary for a seed to pass through the gut of an animal, and the animal, by passing the seed through it, actually begins to digest the seed coat. And this digestion of the seed coat is necessary to allow the seed to germinate. It weakens the seed coat, so when the animal finally defecates it. And this is a signal to the seed, so to speak, to know that it's been transported to another area because its seed coat is weakened. And this is a particularly important phenomenon. Finally, I just wanted to mention that plant seeds are often dispersed by insects. And they are dispersed by insects because the seeds or the fruits, again, have structures or make structures that are attractive to the animals with regard to nutrition. And ants, for example, in New England are particularly important in dispersing seeds. They collect the seeds because of the attached sweet-smelling material, and then they bring the seeds back to their burrows, and the seeds are left untouched. But then if the burrow is not too deep in the soil, the seed will germinate later on, perhaps when the rain comes or when the soil is disturbed. So let me show you now some pictures of different types of seeds and different types of dispersal mechanisms. Oh, better go this way. Sorry, guys. This first dispersal mechanism is one that involves touch. This is a plant which grows right here on campus. There's a big plant of this up by Evans Hall, if any of you are up there. This is called impatiens, and its dispersal is by touch. Here is the fruit of the impatiens. This is the ovary wall you're looking at. And in this particular plant, when the plant is ready to disperse the seeds, the ovary wall begins to dry out. And in drying out, the cells become under tension. They are so much under tension that if you touch it or an animal comes by and rubs it, the ovary wall quickly breaks to release the tension. And this is that same fruit after the tension has been released. And in doing so, the seeds are dispersed upwards, I would say sometimes up to 15 feet away from the plant. So powerful is this breaking of the tension. Here's another example of a plant. This is a salvia, which is a plant that grows in California. It has its seeds already formed, and these are sepals probably on the outside. And if you touch it, you release a trigger spring, which then causes the seed to shoot out and go some distance. Here's an example of a seed which is dispersed by wind. This is milkweed, which grows on the campus. And these seeds, here's the fruit wall on the outside. These seeds have this parachute, which allows the wind to pick it up and to carry it some great distance. Now here is a seed or a fruit that some of you are familiar with. This is mace. This is the material that is used as a spice for mace, and it is an extra layer of cells which is particularly attractive to animals that this plant forms, and in forming it, it allows, and it probably is a pretty large animal because the seed is a pretty big seed, but the animal is attracted and perhaps likes the taste of this extra structure and disperses it. Now, some seeds, especially those dispersed by birds, tend to have contrasting colors for the birds to key in on. And here we have a seed in which the extra tissue produced is red in color, and the seed is the black part. The bird eats it, often for the red color, and then flies away. Here's another one in which there's contrasting, although the material which is red is the ovary wall and the seeds are black, again to make a contrast. Here's a very unusual one in which the seed has a sort of ultramarine color on it. Why would a seed produce this color? Must be some organism which is particularly attractive. This is a tropical plant. For those of you who come from Miami, you may have actually seen this. It's called traveler's palm, but nevertheless the seed has this beautiful blue ultramarine color. What would have been attracted to it? And finally, I want to show you a seed which is often dispersed by insects. This particular seed looks like eyes, and the insect doesn't really care about the color of this extra tissue. In this particular case, it's white. The ovary wall is the sort of reddish tissue on the outside. This seed then is carried to the burrows of the animals, and in being carried to the burrows, it is then the white tissue is consumed, and the black seed is left intact, and the animal then doesn't have to get rid of it, and the seed has a nice place to germinate. So these are dispersal mechanisms, and the dispersal mechanisms are an important aspect of the plants. One mechanism that I want to mention to you that all of you are familiar with, I'm sure, has to do with the ability of seeds to stick on your pants leg or on your socks when you walk through the grass or when you walk through a forest. These seeds have barbs on them, little barbs that allow them to be transported on the fur of animals or on humans. These seeds were looked at by a man, I think in the 1940s or early 50s, who noticed that he could continue to put the seed on, pull it off and pull it on his pants leg, and it never seemed to tire. Whatever was holding it on, it didn't break, and they could do this repeatedly. And when this guy looked at these barbs under the microscope, he discovered that these seeds had little hooks, and that the hooks tied onto his material, and this was the basis for the discovery of Velcro. Velcro basically came from the guy making this observation, and this is an example of the field of biomimicry, which is using nature or nature-inspired designs to help humans to design things that are of use to us. Finally, what I want to do is to just talk to you briefly about germination of the seed. When the embryo is mature, I'd like to talk to you about germination. So here we have this seed, and the seed is buried in the soil. So here's the soil. We have this seed buried in the soil. Now, this seed has a problem. It has to get up through the soil to the sunlight. It can use its food reserves to give it a chance to get going, but it has to get up through the soil. And usually germination is considered to be the most risky part of the life cycle of a plant because there has to be enough water, the seed can't be buried too deep in the soil so that its food runs out, and it has to be able to get to sunlight before those food or nutrients are exhausted. So think of it now. Here we have this seed where the embryo axis has to grow up to the soil. There could be lots of things in the way of this seed. There could be hard soil. There could be a rock. How is the seed going to maneuver or manipulate around these various obstacles, potential obstacles in the soil? Well, the very first thing the seed has to worry about is to protect its meristem. The meristem, we said, is at the very tip of the seedling axis, and therefore, as the seed pushes up through the soil, or the seedling pushes up through the soil, potentially that meristem or that shoot tip could encounter a rock and be damaged. So how is it that the plant prevents itself from being damaged? How does it prevent that meristem, which is a very sensitive part and a very important part, from being damaged by rocks or some other obstacles in the soil? And this is the truly interesting part. I want to show you a seedling, and you're going to see this in the lab. Please take advantage of the fact that you're going to see this in the lab. Here is a seedling which is germinating. Let's say this is the soil level at this point. If you look at a seedling which is germinating at the soil or below the soil, what happens is that frequently these are the cotyledons. Cotyledons. Here would be the shoot tip or shoot tip meristem. And notice what this seedling does. This is the hypocotyl. What the plant does is it uses the hypocotyl as the battering ram. It protects its shoot apex. So what's pushing up through the soil is the hypocotyl. It's pushing up through the soil and thereby keeping the apex or the shoot apical meristem safe. And then once it gets up to the soil surface, if we made for some time, and you'll see this in the lab, what would happen is that it would eventually straighten up and grow so that the meristem now was at the tip. So germination is an important aspect. It's a very interesting part of plant biology because there are a lot of different strategies that are used by plants in order to increase the chances that the germination will result in a successful establishment of that particular seedling. Finally, what I'd like to do is just briefly draw for you a mature seedling plant and put some landmarks on the seedling plant, landmarks that we're going to be referring back to for the rest of the lecture. So let's make the axis here. So we're going to draw the plant axis. And I want to put some landmarks on this plant. Many of these landmarks you know about already. The first thing I want to do is to put the landmarks of the structures that you're aware of. This structure here is a leaf. And the leaf is divided, as we will talk about in more detail later on, into two parts. Into a flat or somewhat elaborated area, which we call the blade, and then a narrow area that can be of varying lengths that attaches the blade to the plant itself. And we call this the petiole. This point of attachment of the leaf is given a special name. We call this a node. So on this plant, which I brought in to you here, you can see we have lots of leaves arranged on the axis. In this case, the blade is quite flat and the petiole is quite short. Each of these places the leaves are attached is considered to be a node. So if we have another pair of leaves here, as you can see on that plant, we would have another node. Now, as you might guess, we can't resist naming the region that's between the two nodes, and this is called the internode. Internode. Sorry, get this R here for you. Internode. Only a couple of more things to put on, and then we're through. At this end, we would have the root apical meristem. And at this end, the shoot apical meristem. And now what I would like to do is to just focus briefly on a node over here. So we're going to look at a high power of a node. And if we look carefully at the high power of the node, what we find in the node between the petiole and the stem is we find a bud. This region is called an axil. An axil, the region between the petiole and the stem. And therefore, these are called axillary buds. And what do axillary buds become? Axillary buds become the branches. So when an axillary bud develops, the branches grow out, and they grow out from a node, because that's the point where they originated from. So basically, that's the terminology of a shoot. Let me now show you a few slides of this. Oops, better go back here. Okay. So this first slide is of a plant that we talked about, and I want to just point out to you the important parts: the blade, the petiole, and the vegetative branch, which was an axillary bud. At each end, you have the shoot apical meristem. At the other end, you have the root apical meristem. Now, when the seed germinates, an important point in the germination of the seeds is that in the dicots, the eudicots, we have the hypocotyl and epicotyl bending. But in the monocots, we don't have this bending which goes on. And so in the monocots, the way they protect their meristem is not by bending the axis, but by putting a sheath covering up the apical meristem. And that sheath, which we're going to go back to later on, is called the coleoptile. And I want to just show you it now, and we're not going to talk about it anymore for a couple of more lectures. But I want you to know where it comes from. When the seed germinates, again, notice the hypocotyl hook is what's pushing against the soil. And once this hypocotyl hook gets to the surface of the soil, it straightens out, and the embryo axis now becomes a straight axis. And this last slide will show you the hypocotyl hook. This seed was buried very deep. Actually, it was kept in the dark, which the plant thought meant it was still in the soil. The hypocotyl hook is attached over here. Here are the cotyledons. The apical meristem would be inside. And then once you give it light, making sure the plant thinks it's above the soil level, the shoot axis straightens up, and the cotyledons, which are here, are almost used up. Okay, have a nice weekend. We'll see you on Monday. Come up and look at the embryo if you want.